take our DNA molecule, we bind to the promoter region, RNA binds, it runs along and makes our mRNA. Okay, we build on the 3' end, we elongate the mRNA. The mRNA then gets translated into a protein. So we have our RNA, a ribosome binds, ribosome binding site, and it goes along until it reaches MET. Then AUG comes in, MET comes in, and forms our N-termini, and then we elongate that out until we either reach a stop codon or it falls off the mRNA. Okay? So we have 3 diseases here, 3 different things. The first one is cystic fibrosis. What happens in cystic fibrosis?
An amino acid is taken out.
Yeah, so what happened? So that's the consequence. So what happened? We have an amino acid taken out. So we have a protein channel, and it condenses down. It becomes a little bit smaller. Just a tiny bit smaller. And so we talked about cystic fibrosis in fall quarter. That was the disease where Europeans have a higher extent of it, but it also provided resistance towards brain just died. Typhoid. Provided resistance towards typhoid. So when that becomes a little bit smaller, now the ions aren't going to be able to move through, and you get a mucus buildup in the lungs. Hence the physiological problems. Okay? So a small change, big consequences. All right, what about sickle cell anemia?
An amino acid was changed.
Amino acids changed. So what amino acid?
A glutamate to a valine.
So we went from glutamate, which is a charged amino acid, so hydrophilic, to valine. So valine is hydrophobic. So we went from this shape to this shape. And now we're no longer able to carry as many oxygen molecules. Hence the consequences of sickle cell anemia. Single change, substitution, gives big consequences. The last one we looked at was hemochromatosis. What happened? What happened? They replaced it. So we had a substitution. Right, so we have a change in the amino acid sequence. So if you have a change, can enzymes recognize it? No.
No.
So it's not going to get broken down. So you're going to have a buildup of blood. Anybody know how they treat this disease? Bloodletting. Bloodletting. This happens to be the one disease where leeches work. So this disease happens to be fairly common in the royal family, especially the French royal family. And so turns out the French royal family discovered this, and they had bloodletting or leeches. It cures them. It actually works. So then all the rest of the people said, oh shit, this works on them. This must work on my disease. No, it only works in hereditary hemochromatosis. So it typically only shows up in men for what reason? Talk to Sharon. Why is it only show up in men?
Some nucleotide.
That's when it does show up in women.
When?
Yeah, menstruation. So typically it doesn't show up in women until they go through menopause. In men it shows up quite early because they don't bleed. So today, how we treat this disease is you just donate blood.
Donate?
Donate blood. Every month or so you just go give some blood. Life is good.
What about the leeches?
Sure, you can do that too.
How?
I don't know.
Leeches circulate the
Leeches
blood.
out. Do bloodletting. It actually does work. This is the one disease that does work, but it's just a substitution there. Okay? So, what was our goals today? DNA. Knowing how to assemble DNA, the structure of DNA. What is a purine?
2 rings.
2 rings. Pyrimidine?
1 ring.
1 ring. A binds to T with 2 bonds. G's bind to C's with
3
3
bonds.
bonds. There we go. And so we now know how to structure DNA. Then we want to understand transcription. Okay, so we build, RNA polymerase runs 3' to 5', building on 3 prime in, so we build 5 to 3, okay? We build 5 to 3 because we have that hydroxyl. The charged nucleic acids with those phosphate groups come in and they are on the 5 prime end, so they have to come in and bind to the 3 prime end. That's what we build on the 3 prime side. So lastly, to kind of recap translation, which we should have a really good understanding because that's what we did in lecture all week. Right? So we're going to build those amino acids using those amino acid chains using the ribosome. We're going to build N to C terminal. We build on the C terminal. Okay? How do we feel? All right. So make sure that the overheads are wiped up. Come on, turn them in whenever. You know, if you want to hang out and finish stuff up, you go for it. I'm good.
What about the
Wait, do
promoter?
we have to write? Do we have
We'll
to?
learn all about the promoter you're coming up with. The promoter just is where it tells it where to bind the
Do
transfer.
we have to finish this in the
We
lab?
turn it in. We turn it in online
just
now.
turn it out
This
online,
weekend, right?
Yeah.
Also, why do you think that Baltimore is different than Seattle?
Because it is different.
I was wondering this.
It's not the same university.
Okay.
Okay.
Well, yes.
So, different opportunities, different experiences. Doesn't have a graduate school attached to it.
The first,
the
last
Doesn't have
page of the
research
first-
labs attached to it, so it's not an
Last
R1.
page of the
It's a
of the
community
first
campus,
packet.
so you don't have
cystic
the same
fibrosis.
classes,
No, not
same
the last one, I'm
dynamics,
sorry.
and the degree offerings. are
Oh,
the
This
same.
one.
oh, that
So
one.
the best
Okay.
example, because I know
But mine
really
is
well biology.
different.
UW Bothell biology does not require OChem.
It doesn't matter.
UW Seattle biology, you have to have
Because,
OChem to be able to get into the biology sequence. So it's very different.
Yeah, because I want to do my degree there.
You need help like
Yeah, it's very